Okay, so good afternoon, everyone. As some of you are familiar with how I start these sessions, I'm going to start introducing myself and then the topic of today's session, and then Anji. So my name is Isabel Ramos, and I'm part of the executive committee of PARSUC. I mean, you might have been familiar with this already, but PARSUC is a Portuguese association of researchers and students in the United Kingdom, an independent and non-profit association without political affiliation, which aims to represent the interests of Portuguese researchers and students in the UK. We function as a network to promote the research done by the PARSUC members and to bring together our community and researchers in Portugal and in Portuguese-speaking countries. I want to welcome everyone and thank you for registering for this ENVOLVE training program, a partnership between PARSUC and the Fundação Carlos Gulbenkian. The webinar is being recorded for educational purposes, so if you don't want your cameras on or your microphones, please feel free to either leave it on or switch it off. I also want to let you know and please ask you to fill in a feedback form, which will be sent to you to your emails within the next few days, and this will help us with future training sessions. This is the third session of the ENVOLVE training program, and we will hear about translation of research. And the session today will include a presentation by Dr. Anji Miller. I would encourage you to use the chat button or to ask any questions during the conversation. As Anji said, this is more an informal conversation rather than a lecture. So translation research is crucial to innovation. However, this activity is dependent on the ability of researchers to secure funding through funding grants and awards. And the Strong Translational Research Project will not only address an unknown aspect supported by a clear hypothesis, but also articulate the path to the solution and how this will be translated to the real world. So this training session aims to highlight and discuss the key aspects of a robust translational research hypothesis and the key requirements for the proposed research to validate the given approach. It's a great pleasure to introduce you Anji. Anji is a technology transfer professional with over 15 years experience of working in an academic and non-profit sector. Her current role as a senior business manager at LifeArc involves working with academics and charitable establishments to identify and cultivate early-stage healthcare technologies. As a passion advocate for the technology transfer profession, Anji leads the LifeArc Autumn Technology Transfer Fellowship Program, a scheme that helps life scientists enter the technology transfer profession. She has extensive experience of IP and contract management, licensing, business development of healthcare technologies, and development of knowledge transfer policies. And Anji, the floor is yours.
Thanks, Isabel. Thanks everyone who's agreed to join this session. I'm quite excited to actually articulate some of these points, because I think what was really nice was to see a program that was going to drill down on some key aspects that sometimes are glazed over by other researchers, and even quite seasoned researchers can sometimes get this wrong. So let me just share my slides with you. Hang on a minute. Can you see them yet? No?
No, not yet.
Okay, let me just come out of the... Right, I'm just trying to see if I can see the share.
It should be at the bottom of the Zoom. If not, I will make you a host.
Hang on a minute, let me see. Will it allow me to do this?
It's green down, share screen.
Yep, I've got
Can't
it.
you see?
Okay, yeah, I've got it now.
Thank you.
Okay, and then I'll just go into presentation mode so you can see. Can you see it now?
Yeah.
Yeah. Okay, and let me just go back to the beginning. Okay, I won't bother to bore you again with introducing myself. I'm hoping that this session will connect really well with the previous 2 sessions that you've had, but also with the other sessions that are to come. So I've tried to design it in a way that it would just fit in. But there are 4 key take-home messages or key aspects that I'd like at least that you will be enlightened on after we finish talking. And the first is really, when we say translational research, to really define exactly what we mean. Because the thing is, sometimes it's not quite the same, and it does vary depending on the discipline. Also, for you to really appreciate that every technology has a completely different development path. They do overlap, and there are some commonalities. But the thing is, it's when you are involved in translational research, whether you are going to take your technology all the way or even part of the way, it's important that you recognize the development path. We'll then start talking about what you should have in a strong translational research proposal. And something that LifeArc does a lot, and is a major part of my role, is looking as a funder, exactly looking at the key aspects and what's on the wish list that we'd like to see. Who am I? You've already had the introduction, but basically my background is molecular biology. So it was gene therapy for cancer. That was my PhD area, and the area that I did my research in for quite a long time before I left the lab. And then also trained to become a technology transfer professional. And when I say technology transfer professional, it means I help researchers to get their technologies to a point where it will be supported or taken to market by another party. And whether that's through funding, advice, or actually development, we do all of that. In the next couple of slides, I will just enlighten you to what LifeArc does, because I think it's quite crucial in what we're talking about to understand what a researcher does and what a lot of the funders do, and how they connect. But also sometimes you've got an establishment like ours that conducts research, collaborates, advises, and funds. So we do the whole spectrum of activities. So LifeArc is based in London. We do have 3 sites, but we're a self-funded charity. So the thing is, we don't have any particular thing that we need to achieve each year apart from the goals that we set ourselves, because it's our own money and from our own profits. We work with companies, charities, research organizations, and academic sciences, but our overall aim is to ensure that medical innovation benefits patients to get the technology to the patient, so where it needs to be. So everything we do, that's the main mission. We were established in 2000, so it's quite some time that we've been around. We have a good, decent legacy. It's over 20 years. And because of the success that we've had with a lot of the technologies that we've developed, obviously, you can see what we spent to date on the translational activities. But the thing is, like, we have over 200 employees, and we're in 3 sites, all based in the UK. So that's in Stevenage, London, and Edinburgh, where we have our diagnostics. Our research labs are in Stevenage, and where I am at the moment, which is in London, that's the main offices for our technology transfer department. What we do is focus on translation of research. This is why I wanted to give you these couple of introductory slides to just put things into context. We help researchers, whether they're very early, as you can see in this depiction here, this image, to try and move the technology further along the development pathway. And I'll talk some more in the following slides on what we mean by development pathway and how it can differ and what could be involved in those and the questions that, as a researcher, that you would ask yourself. But we do this in the aim that we will fill the gaps, because what you'll find is when you start research and you start looking for funding, you'll see that there are gaps in funding, gaps in development. And unless you have an entity like filling those, it is where it's a big challenge. So you could be working in an area It's great that you're an expert, but to get it off the ground, to get the funding to support it, is really quite difficult. And even if you do get some funding, it's the subsequent funding after that that you find quite difficult. But what we do at LifeArc is to try and help move things along. And that could be by building on any of these aspects, for example, the rationale or the hypothesis. You know, if you're searching for any particular small molecule, the chemistry that's involved in that, the medicinal chemistry, or maybe preparing a diagnostic ready for market. Just looking at the expertise that we have within LifeArc to be able to do this, and the sort of expertise that you are going to have to develop as you develop your research, and if it's translational research in all these areas, or seek the expertise to help you move your technology along. That would be translational advice. For example, like myself, our teams are experienced and skilled within intellectual property, managing portfolios, liaising with third parties, because one thing you will find that translational research is very much collaborative. It's not where you just do it by yourself, as in basic research. You do require input of other expertise that you won't necessarily have. And that's the nature of translational research. We also have our labs, as I mentioned, that we have expertise in antibody humanization, diagnostics, but also translational research in any aspects. And we will work with collaborators because we have a model to do that. And then the third aspect, which is what we're going to touch on at the end of this talk, where it's translational funding. We provide translational funding, and we have several schemes that will help take the technologies from relatively early right the way through to phase 1, 2. And also we have like seed funding. And then what we are looking at in the future, what we'll also have is venture funding. So enough about LifeArc, but the thing is I wanted to put that into context on what we do, my world, working with translational research, and how it touches and impinges on this aspect, your hypothesis and translating your research. When we talk about translational research, I think it's important that we're all on the same page, what we mean, because the thing is, working with academia for many years, I've had so many researchers who say, yeah, I'm translational. And it doesn't mean that you're conducting basic research, which is really quite specialized, and maybe obtaining a little bit of blood and somehow incorporating the cells in your experiment. That's not translational research. There is a big difference. When we talk about, let's talk about the different types of research and how they relate to each other. Basic research, that's the fundamentals, and everything uses that, because that's the information to understand how these things connect. So it's really looking how everything ticks over, how these cells generate. Translational research uses the outputs from basic research, and that's where it relates them to the human body. And then when you look at clinical research takes it even further. So you understand it in the human body, but the thing is, what does that mean? Can you cure that fault? Can you correct that function? Can you enhance that function, or can you dampen it? Or can you detect? Can you measure as a diagnostic? So all three do interrelate, but it doesn't mean, for example, you're just looking at pancreatic cells in isolation. That's basic research. It's not translational research. And it's important that, when you apply for any sort of funding, or you have any sort of hypothesis, you're realistic and understand when it's still classed as basic and when it's classed as translational. There's nothing wrong with the two, but the thing is, it's when you badge something that is basic as translational, it will go against you when you apply for translational support, because the thing is, they are really looking at how does that impact the human body, and how would that impact the patient group relative to the technology? Here, this is just an image that I, to get the points across and to really expand on how you would think as a translational researcher and what you're doing at each key stage to understand how they relate to each other, and how really it is you can't get a technology to a patient without understanding and using the information from all three. So for basic research, you're looking at, what can you learn? For example, it could be cells, it could be a particular function, it could be a particular disorder. When you're looking at translational research, you're looking to see what's gone wrong, because it's in the wider context. It's not just that small function, it's how, the chain of events, how does that affect other things? And how can that, in any particular patient group, what could have gone wrong and what other activities, are really playing a role in it, because it's not just one chain of events or one pathway, it's usually multiple pathways. Once you understand that in translational research, then it's really where you start going into, okay, how can we fix it? How can we amend it? Or how can we fine tune? And this is where you start to look at correcting syndromes, correcting disorders, and it's the clinical trials. The image at the bottom was really just, again, another example by putting in, for example, basic research will look at mechanisms of diseases. So you use the techniques that you'd use, are cell culture systems, animal models, use lower animal models, not the higher. And then you're also looking at the molecular techniques that you would use for PCR and things like that to amplify. When you're looking at translational research, you are looking then biomarkers, which is a marker of any particular activity within the body. The targets, for example, if it's a particular target cell receptor, because you've understood from the basic research where there is an issue. And then you're using methods, for example, like sequencing. And this is also where you'd be using higher, so you non-human primates may possibly in translational research, particularly when you're putting together a package, which is a preclinical package, the package required by the regulators to allow you to conduct clinical trials. Then you've got clinical research, where you're looking at managing and really looking at safety first, which is phase one, which is I won't go into too much details about this, because the thing is, that could be a webinar in itself, the different phases of clinical research. But the thing is, you're looking at safety, then you're looking at efficacy, so how well it works. But the thing is, at that stage, you're looking at humans, it's in humans. So it's clinical research. Is that okay so far? Are you guys, you're with me? Yeah?
Yes,
Ooh,
yes.
I can see, Pamela, you've got your hand up.
Yes, I have a question regarding this. Because, for instance, if we think, I'm more on the side of the basic research, okay? And for instance, if I will give an example to be simpler, to be more simple. For instance, CRISPR-Cas9, okay? CRISPR-Cas9 is a technique. Whenever it was being studied, I think it was considered a basic research, okay? And then it became more of a tool, more of something that can be applied. So considering the explanation that was given so far, so how would this work? So because I became kind of confused. So at this point where we are at now, can we consider it part of a translational research? It's not the basic anymore, because now it's more of a tool, right? That is being applied in several, I'm not, yeah.
The tools aren't mutually exclusive to the phase of research. So the thing is, in translational research, you would still use cell cultures, you would still use cell culture systems, but the thing is, it wouldn't be the ultimate, because the thing is, you are translating, you're translating what you're learning in a human system. It's closer to the humans. basically, as you move from basic to clinical, you are getting closer and closer to humans. So when you're conducting, the difference is your approach, not necessarily just the tools, because the thing is, in basic research, it's usually in isolation. So it may be these cells in isolation. You know, basic research, a lot of the time you're using Drosophila. It's because that's the system that you can use to learn a lot more, because you know how close it is to the system that you're studying. So it's not just the tools. With CRISPR-Cas9, yes, it does hold a lot of promise, but the thing is, because it's derived from basic research, this is where, again, there's an issue, and it's going to be the challenge, because so much is unknown. You know, how CRISPR-Cas9, the way it is probably not going to be the tool that would be used in humans. It's probably going to be something fine-tuned, because the thing is, you have to be able to know exactly what you're doing and what the knock-on effects are. And then also, you have to be safe, because the risk-benefit has to balance when you're doing anything in humans. Hence why it's a long path to get in humans. Does that explain anything? Is it more clear?
Yes, thank you.
Yeah.
I mean, Anji, what would be an example of something that perhaps LifeArc has funded that involves, for example, CADS as a technology?
That's translational? That's
yeah,
translational?
Just to be...
Yeah. For translational, there's a lot, particularly one of the funds that I work a lot with, which is it's for rare diseases, gene therapy approaches. For example, AAV9, because the thing is, that is the viral system that has had the most de-risking and has gone the furthest. Particularly, you have gene therapy for SMA, but the thing is, what we're looking at through the philanthropic fund, which is the fund that funds rare indications, this could be something simple where it could be a disorder that only affects like 5 people. But the fact that it's gene therapy and it's possible is that because it could be where the basic research has been conducted where they know what's gone wrong. The translational research is where they've proven the concept of what they've found in the basic research to say that, for example, it's not enough of this particular protein. So when it comes to the putting together a package that you're going to test it in the clinic or in patients, it's really where you are getting all the data to show that it's safe to actually switch on that protein that's not being produced, and it will cure any particular indication. At the moment, one of the largest, and it's in clinical trial, it's basically, it's a gene therapy approach, but the thing is, it's using the patient's own T cells, because they know that in this particular indication, it's aplastic anemia, the T cells don't function well. So what they are doing is taking T cells, actually upregulating them, these are patient's own T cells, upregulating them and getting them to work correctly. And then eventually what they'll do is they'll give them back to the patient. It's a gene therapy approach. And rather than adding something exterior, it's quite clever, because it's getting the actual patient to cure themselves by actually getting correction of the function. But it's not by adding a gene, but it's a gene therapy approach. But the thing is, it is where it's just getting, because the machinery is there, it's to switch it on and make sure that happens. And that's an example of a translational project that it's in clinical trials. So it's at the cusp of translational clinical, because the clinical is still translational anyway. But the thing is, it is where they are proving safety. And then because the numbers are lower, again, another aspect is when you have a rare indication, you can get to market quicker, because you're not going to have the populations that you would normally have. So the proof of like safety and things like that are usually more condensed and with smaller populations. So it can happen much quicker. The regulators, once they know it's safe, it can happen much quicker, because you need this to happen, because you've got patients dying, you've got people dying, these indications, and it's not attractive to anyone else. Is that okay? Yeah?
Miller, sorry, I've a doubt. Can you back the...
Yeah,
Yes. It is possible to do a translational results without a basic research?
is it possible to,
Is possible do translational research without do basic research?
for a researcher to do that? Yes, it's possible, but it's not possible in the fact that someone would have done the basic research, because the basic research provides the knowledge that you'll have to conduct the translational research. You would have to, it would be maybe in publication. Say, for example, the reason why I said yes is because you could design a translational research project. It's translational because basically you're not looking at just the main function of these particular discrete organs or cells. You already have enough information and data from literature to know that, okay, we'll keep it simple. This particular disease is caused by a particular protein not being produced. There's data, there are publications clearly stating that, and these are peer-reviewed publications with the data there to say that it's caused by, for example, basically a gene that's not correct. So it leads eventually that you have particular cells not functioning right, and you don't have a particular protein. You could start and design research around the fact that this is known, this is state of the art, this is known in that area. And the project that you're designing is translational. However, you didn't conduct the basic research because it's already been done. But you will still need that knowledge from basic research to conduct translational research.
Yes.
Does that make sense? Yeah?
Mm-hmm. Yes.
Okay. I can't see. And no more hands? Yep, that's okay. Okay. So I just wanted to explain exactly what we mean by basic, translational, clinical, and how they interrelate, because it's important, and we've touched on it already, when you are devising a hypothesis for your research. So when basically, this is almost like the blueprint for any application that you will make for funding for translational research, or any funding, in fact, because it's the concept, it's the idea that you are testing and proving, and that this will be almost like the key validation data of the approach that will be developed as you move along the development pathway of the technology that you have. And it does use all the information that we've discussed before, but also it is where it uses good laboratory practice, where, you know, there are some key aspects that you have to have there. Otherwise, when your research or even any publication is peer reviewed, it will be devalued and not taken seriously if it doesn't involve some of these key aspects. So, you know, having a sensible hypothesis it's key. What is it? You know, it's more or less it's when you break it right down, regardless of the discipline. I know all of you are life sciences, medicine, and everything. It's more or less the same. It's a concept or an idea that you're testing. And if you break it down for when you first entered the lab, many moons ago, when you first started to do experiments, you were testing something. And the hypothesis was the concept the idea that you were testing. And what you do is you prove it through research, and these are controlled research and experiments. So the thing is, ideally you want to change one parameter at a time, or you have one variable at a time, or everything is controlled so that even if you do change others, everything is controlled against that change that you're making. It's not, you can't have just blue sky thinking and it be groundless. Even when you start basic research, you will do literature review so that you make sure that you are not repeating something that has already been done and has already been proven, because it would be a redundant exercise. Yes, you've got great data, but you'd already be looking and just, unless you need to, you wouldn't really be repeating someone else's experiments with nowhere else to go with it. It's really where, if you repeat it, it's really to make sure that you can reproduce the results and the conditions that have already been published. And that hence the reason why people publish, because that's information out there for you to use to build on. But whatever hypothesis you have will make sure that it takes into account the information that's already there published. You do use that. You may even talk to experts, because although a lot is published and it's out there, a lot is know-how from those who are working in the field, in the area. So it really is where it's key that you get the hypothesis right, and that when you are applying anywhere, you put things into context. There's a story around the hypothesis, how you came up with it, and also why you're taking a particular approach, why you're using particular techniques to actually prove it. These baubles here that say who, what, where, when, how, basically, if you had to break it right down to basics, this is what any reviewer or anyone who's looking at any sort of approach will want to understand. And by doing that, you will cover all your bases to really support and justify your hypothesis, but also justify the strategy that you are using to prove it. Most of the time when you're in translational research or research, yes, you have a hypothesis, but they don't use the term hypothesis. What they will say is proof of concept data. And it's remembering that when they say that, the concept is the idea, what's been proven, and that's usually the key data that you can move on to the next stage. You need to have that. Or you'll get sometimes with a review that this needs to be validated, or that needs to be proven, or there are particular questions. It's because these are looking at the data that you have there, and there are still some holes, there are still some unanswered questions before you move on to the next one. So if you had to look at some of the key components of a strong hypothesis, is that you can have dependent and independent variables, and these are the parameters that you would have like in an experiment. So you have to have a control against anything that you are varying or changing. You have to be able to prove the hypothesis. Otherwise, it doesn't make sense. If there's no way to prove it, it's not really a hypothesis in that respect, not for research, because it's something there, but no one will ever be able to even test it. You have to also be able to show that it's not true, or it's not. So you have to be able to, it can't be that it's designed, the experiments are designed to show what you want. It has to be able to show that if it's not the case, that they are negative. And then, like any good laboratory practice, as I mentioned before, it has to be reproducible. So obviously the multiples that you would have when you conduct your experiments. But the thing is, for this to be taken forward in any shape or form, regardless of when you decide to publish, someone else should be able to follow what you've said and prove and find the same thing. Otherwise, it is where it's a dangerous ground, because you would have something that may be published, but then again, it will be that you will have a follow-up of publications, data, and everything that will say likewise, and it will discredit not only the research that you've done in the paper, but also your research and your approach. Okay. Just touching on what I've mentioned before, what makes a good hypothesis. Solid question, the research around the area, so understanding who's done what technologies in that space, what's been published, what are the other theories. Usually when you're in an area, you've got, sometimes we think this pathway is involved in whatever, understanding that. And that's something that you have to articulate in any application that you have, so you understand. It's almost like a review of that key area where your hypothesis is. So you really do have to conduct your research and speak to others to see and bounce the ideas. Trusted individuals, you can easily do that. But also you have to be able to test it, as I mentioned before. And this is the key part of the beginning of any funding application, because the thing is, this is where you set, like, you know, you've got the title, but when you start talking about what you hope to achieve, it's because you are supporting a hypothesis or you are going to completely test it. Right? It depends on how far, and we'll go into this with the technology readiness levels, so how mature the research is, what you're trying to prove and what you're trying to test. As I mentioned before, proof of concept data, when they use that terminology, they are talking about hypothesis. So it's proof that the hypothesis is as outlined. So this is the data that you've really obtained once you've done those initial experiments, and you're building on that. So each time you've proven a certain part, you're building on that. And that's the proof of concept data that forms a whole body of data that is used in translational research as the foundations, because that's what you're building on. Does that make sense? Because that's a key aspect of what I wanted to get across in this session. Hypothesis and how important it is, but also some of the key attributes of a strong hypothesis. Yeah?
Yeah.
Yes, Pamela?
Yes, so there are two topics, two things basically that you talked about. One of them is the reproducibility of the work you do, right? That is I mean, every researcher, right, should guarantee that the work he does is reproducible.
Yes.
But we all know that nowadays that is a big problem.
Yes.
Right, nowadays that's a bigger problem. So I would like to hear a bit more about it. What do you think about it, about this issue?
It's key. And there was, some years back, there was a publication that said, I think it was over 70% of published research, when they had a look into it, a lot of it couldn't be reproduced in the conditions that were outlined in the publication. What you've got is journals are trying to police it more to ensure, so they will probe and ask more details, the research methods. Yes, you can understand why some are being slightly more cryptic, because they may be competing with another lab and they're working in a similar area. But the thing is, when someone is totally fabricating, or what they found is it's not appropriately controlled, and their successful data is almost like a fluke, so it's luck, it's not great. Because the thing is, there's a certain level of integrity when you are conducting research and publishing. Obviously, there's a pressure to publish, because as an academic, that's how you say you're an expert in a field, and you show, you mark your territory and you progress and you get the grants. But it's not a good thing, and it's global. It's a problem globally, and it's at the highest level as well as the most basic level of research, because you have some very high profile labs that some of the things they claim were not true at all when you look into it. The only thing that I can say is that you in yourself have to make sure that the research that you conduct is appropriately controlled. And if you have any doubts, do some additional experiments to support it, or explain why it's that way, because you will be challenged at some point, and it may be directly when you try to publish through peer review. It may be once it's published, or it may be, worst case scenario, when you think that you are a leader in a field and it affects not only you, everyone in your lab as well, because the thing is, it's tainted. You don't want to have that, but it is a problem, and it's recognized as a problem. I think it is the only way that that can happen is there's a movement that are really going out and trying to randomly pick pieces of work and try to reproduce them, and they will publicize that. But the thing is, I think it's the only way is to make sure that you're not one of those individuals, because you won't progress. It's short-term success. You can't build on it because it's not the way it is. So it won't get further down the line.
And also there's a second topic, a very quick one, just because I would like to hear a bit more about what do you consider as a strong hypothesis? Why am I asking that? Because, for instance, as an African researcher, right, we all know that there's a lot of gap information when it comes to Africa concerning research, right? So there's, I think there was some section before that we were talking about it actually, because sometimes there are some kind of researches that have been already done, right? But those answers do not apply to our region, and so. Yes. There's this need of doing more characterization research, because we are we still need to go higher when it comes to that.
Yes.
So how would you judge a strong hypothesis in this situation? For instance, for African researchers that are seeking answers in their own region, and
I think it is where it's the hypothesis would take what's been published or what's already known for a particular region and say why it doesn't, or it still needs to be looked at to apply to, say, for example, low middle income countries, and whatever setting you would have to say it's still a strong hypothesis, and you're still using data and information that's there. It's just that it's closer to what you're going to prove. Basically, it's just that what they've tested and found does not apply to the region or population of, for example, say it's Africa. How can you have something that doesn't apply to a whole large area of the globe and individuals there? So it's really where your hypothesis would be that to be applicable and useful for this region, you need to incorporate this because of this reason. And you would be building on what they've said, and you would certainly use that. But the thing is, your hypothesis would be all encompassing. So you would be looking at not only the population that they've already found, or regional, whatever they've already found, but also extending it. So it would be controlled. But the thing is, you would be repeating some of their experiments, because your experiments by their design would have that in there. And that's fine. That's good research. And that's also building on what's nice, is that's building on what's there, but it's also showing a lack of foresight, a lack of broader thinking of, for whatever reason, the research that was published beforehand or that was released beforehand. It's important that any technology can be applicable globally. So the thing is, like, for example, the best technologies are the technologies not only that you look where you can apply it to, for example, developed countries, but the thing is the higher income countries, but also in field settings. Because the thing is, for example, if you are dealing with, take for example, malaria, and where I was before, the Wellcome Trust, and probably all of you know about it because it's global, and any technologies that they support, it has to be affordable and accessible globally, so for all territories. So if you are dealing with malaria, which is very much a wealthy disease in some regions of the globe, where it's a real nightmare and a real issue in others, if you have something that basically is a prophylactic, so can prevent you getting bitten by the mosquitoes. So it reduces, almost eliminates the chances of you having malaria. You don't want to have it at a price that it can only be afforded, you know, accessible and affordable to one part of the globe. You want it so that it's affordable and accessible to the others. Obviously, the economics of technologies getting to market, which is another thing that I could go on for days and days about, sets a certain tone. But the thing is, you would want to ideally have a business model that, yes, you can charge it where you have a margin in certain regions, but the thing is, it's affordable and accessible in others, so that everyone, this is going to be, you're going to eliminate this problem globally. That's the ideal scenario. But then, as you mentioned, you have some research where it's just not, they have not thought about certain parts of the globe or certain ethnicities. It's for whatever reason. And if you want to address that gap, you have to make sure that you are aware and know. So the knowledge that you would use is what's already out there, but you build on it and you show why there's a difference and why their technology, I think the onus is not only extending it, but the onus will be on showing why their technology or whatever they're finding has not taken a certain aspect into account. What's the difference? Do you see what I mean? Yeah?
Okay. Sure. Thank you very much.
That's okay. Okay, so we move on to the next part where we talked about translational basic research. We've talked about, you know, having a strong and robust hypothesis, but it's also any technology that you have to know or have an idea, even though you're not going to be involved in all those stages. It's knowing how would you get that technology to, for example, be in that diagnostic or that therapeutic on the market. Pamela, did you have a question?
No, I think I forgot to remove the marks. Sorry.
That's all right. But so here I just wanted to give an example, because the thing is, obviously it varies depending on the technology at hand and where you start. But the thing is, the reason why I wanted to have this here is that you see that it's a long path. It's a very long path, and for a lot of technologies, it can be very expensive. So you've got basic research on the very left. And this slide just shows you some of the key areas that we mentioned. Basic, the early discovery is where you are moving into translational. And then you go, I mentioned before in the talk in the preclinical package, this is the package that's required. This package of, it includes everything generated from basic all the way up to that point that would support your technology applying to the regulators to be able to be tested in humans, which is the clinical development stage. Obviously, next it's FDA. So this slide is talking about the US. So you're talking about the US regulators, and then when you get to the market. And remember, I mentioned before about phases, research phases. You know, here, these are the clinical phases, the key clinical phases that you usually hear. You can break it down even further, but this isn't something we're going to talk about here in this talk. But the thing is, it's something you should be aware of. And you should be aware, this is for example, maybe a therapeutic. For a diagnostic, it would be slightly different, but you should be aware of the different phases, whether you're starting here, whether you're middle here, or whether you're here. You should be aware of the next stage, because when you are planning, developing any sort of translational research, you're moving it over to the right. But the thing is, you have to, and most funders will want to know, have you thought about what the next stage is and whether you'd be involved or who else would work with you, but also who you apply for funding for. They don't want to fund something that's going to stop. They want to fund something that it's translational, so then they have a vision that it would get to the patient population. So when you draft any application, you have to think big. It's not lying, but you are aware, and you're aware. They will ask, you know, when you're going to publish, are you going to protect any IP? Who are you going to work with? Who would develop it after you? How how far are you happy to take it? This is what they're asking. They want to know really, how far along here will you take it, and are you aware of the steps next? Like Pamela asked about, technologies that are not applicable to Africa. If you've got technology and you're saying it's global and whatever, the short sight is that it's not global because you haven't taken into account how it will be used in those territories and for that patient population and the other challenges that population may have. So the thing is, they want to see that you have thought about that. And something I'm going to touch on shortly is that all of this are considerations that you need to have in mind and understanding that you need to have in mind when you are addressing a funding call, which is usually has a remit. So I won't go on to that yet, because the thing is I want to just finish talking about the pathway. Here I've circled basically, let me go back. I've circled here the key translational area. When you're talking about translational research, it stretches from there to there. The rest is like basic to the left. And then it really is where it's clinical. It's clinical trials to the right. But the whole developmental path, dependent for the average number of years, depending on, say, for example, small molecule would be around about 15 years. If it's rare, it'd be shorter, because I mentioned that smaller groups, you can, the regulators have ways to expedite it because you won't be able to do all the testing like you would normally have. So it's shorter, but the thing is, you can see that that's a lot of years. And when you're looking, it's usually it's been estimated to get a drug from start to finish to market, 1.3 billion. So you're talking about a lot and a lot of players, because from phase 1 onwards, usually an academic researcher wouldn't be conducting the research. It would be that it may be taken over by someone with deeper pockets and the capability to fund and sponsor clinical trials. So this is where you may work with a company or some sort of establishment that can actually do that. So it's important to know where you sit within, your research sits within this developmental pathway. And this is for any technology that you're working on. It will have the different stages and different timelines, but you should be aware of what they are. So it's realistic what you're proposing. We mentioned the hypothesis. They have to be realistic. And sometimes they are looking at your hypothesis and seeing like, how would that translate into, they use the term target product profile. And that's basically the product that, would it be an injection? Would it be a tablet? Who would take it? When would they take it? Those are the sort of things that they're looking at. And as a translational researcher, you will have them in mind. Okay. We mentioned the development pathway of a technology. One thing that I wanted to also capture, and this is a busy slide, is because I wanted to get this information across. I've put some links at the end for areas where you can pick up and have a look and read some of there's millions of publications. Once you know what you're looking for and what you need to read, the areas you need to read around, but it's a start from the links that I've put there. And one of the things that I wanted to capture here is that not only do you need to know your development pathway, but have an idea, the category of maturity of your research, where your research sort of falls, because sometimes it's specified depending on what funding call you're responding to. So these are technology readiness levels. And this is a method of saying, for example, it's early research, TRL1. But key things you would have at that stage. And then as you progress, you go along the TRL level. And this slide just captures that. But also it captures in this slide, which I wanted to articulate, because I mentioned it before, is where you have, it's mainly leading by academia. Then it would be a collaboration, because translational research is collaborative, and that's that bit in the middle. And then where you mainly be leading by industry, because it's getting the technology to the market. Yeah. So this slide is to get across that point, but also as reference for you. Was there a question? No?
Yep, from Emilia.
Amelia?
Yes. Thank you. My focus is implementing services, policies, dealing with things that was, some of them, as Pamela said, was already proved in another countries, but in low and mid-income countries, some of these things were not already proved. How does it work? Because there are some things that in high income countries, even being proved that yes, it works like this in our countries is different. So how can I, what is the relationship between like the things that I'm doing, like implementing service? Okay, I know that the palliative care, it improves the quality of life of the patients, but the concept of quality of life is different from between countries. So what is the relationship between this and the translational research? Thank you.
The relationship of, sorry,
dealing with implementing new service to make some policies that we don't have, or try to apply the policies that already have to see how does it works in our country. So what is the relationship between these, or how can it be attractive for funding? Because it's not something new.
No, that's true, but that's still fine. The attraction is that basically the funder will want to see a certain, for example, a certain indication or certain activities take place in, for example, the low middle income countries. They don't want to, why would you have this dichotomy where it's not a problem in one region, but it is in another? So the funder would be slanted towards addressing the imbalance. And that would mean a lot of the time the proposals that would come through, or what they would fund, would be based on technologies, activities that are just for the high income countries and have never been applied or are not even set up to be really flexed to address and work in the low to middle income countries. That's the attraction. And that's the reason why someone would do that. And then also it's really where the reason why you would have companies at the end would be interested in it, in implementing it. It's because those companies operate in those low to middle income countries, and someone has to do the work to get to really translate the findings so far to that setting. So it's still attractive and it's still research. But the thing is, a lot of it would be, sometimes it's really finding that what is being said is not necessarily, this is sometimes where you would find that data is not reproducible from what's already been published, or findings don't quite fit. And sometimes the work is trying to find out why. Is it just bad results, bad research, or is there something that really, there's a difference? And this is why it would have to be controlled very well to do this, because you're working with known, applying it in a setting so that you can use it. So it is still attractive and it's still great research, but it's not necessarily where you're looking something new. You're looking something new, but you're using some known to move on to it.
Okay, thank you.
Yeah,
yes.
And where it comes with the readiness level, it starts with the level that, you know, the data was there at. It's the same in that respect. It's the same. When I said that, you know, when you're conducting research and you need to have knowledge of what's been done, who's been doing what, when you're applying data and results from say it's very specific to a particular region, to another region, it just means that the data and results and everything it's already been done for a region that you're applying to another region. It doesn't mean that it's any less value or anything like that. Yes, you do have to repeat a lot of it, but sometimes when you're using data anyway, you have to repeat some of it to move on, because you have to be able to show that that's fine, and it's not, you don't just take it that that's a given. Otherwise, when you conduct your results, you have to be sure that the variable is the only thing that's varying. So you have to have something that is a known against the variable, and that sometimes means repeating certain aspects of what's already been published or what's there. Yeah, does that make sense?
Yes.
Maybe just to add on that, Anji, if you can maybe talk a little bit about when a proposal is being drafted. And one of the key things is also to compare with a gold standard, right? So if you come up with a new way of assessing something, one of the first things you are asked for is how are you going to compare this? And
why? The thing is, you have, because you would say like, oh, this is great, it's quicker. Okay, it's quicker, but they would not only be looking at, okay, it's quicker, they would be saying, why would you do this? What's wrong with the existing that's been used by thousands, possibly millions of researchers? What's the fault? What's the progress? What's the advantage of using this new method or new assay? And you can't just say like, oh, it's quicker, because that doesn't fly. They really want to know, because you need to validate that. And this is where, when you validate something, it's been measured enough times that people take it as like, yes, that's good. That's the new. For example, when CRISPR came out, it's really where people had been using zinc fingers and talons for ages. I mean, the thing is, like, even from when I was in the lab. So the thing is, what was the advantage of CRISPR? It's really where you have to show the advantage, and the comparison would be with the gold standard or the platinum standard that everyone's using. But also sometimes you'd have to go old school and include something further back and really show, okay, this is the advantage. It really does perform better. And it gives you this, or it gives you additional information that you would not get, or it's more specific. Because one of the things, as you become more translational, not only are you fixing something, you have to be careful that when you fix something, because the human body doesn't work in isolation with certain functions, you have to be sure that you don't correct something and it offsets something else, because the body compensates a lot of the time, or it's a bigger, this whole issue is bigger than what you think. So it's really where, this is why anything you bring in to a different setting, different indication, you have to make sure that all the parameters are there, all the controls are there, so everything that's been done before has to be repeated and alongside whatever you're changing or you're introducing. Yeah.
Okay, thank you very much.
Okay. And with the TRLs, I think this is just where you will hear this. You won't so much hear TRL, the term TRL in industry, because they don't really use it that much. It's in academia that you need to be aware. And then certain funders really do use it a lot. So it's being familiar with the jargon, being familiar with what they would expect. And this is also when you know that you would be classed as moving into a different area, when you can see that, the basic principles observed, you've already got that. Technology concept has been formulated, you know that you're going to be classed, or what proposal you have is going to be classed at TRL2. Sometimes funders, when you have a look at the eligibility criteria, they will say that they don't want to fund anything at TRL 1 or 2. They want to see it at least that those two areas have been addressed. So you know that whatever your proposal is and the data that you've got to support your proof of concept / your hypothesis would need to fit those criteria already. Yeah. Apologies that this is so fuzzy. It's really, I had to enlarge the image because it was just too small. At my age now, it's really where you can't be dealing with these diddy writing, but it's made it a little bit blurry. Okay, so if you're happy with that, now what we do is we'll move on to the last stage, which is what I want to touch on. And this is with regards to, okay, your funding application. You've got all that knowledge, all that information. How are you going to ensure that you formulate a strong funding application that's going to be supported? You know, I touched on some of the key things and some of the things that really annoy me. It's one, if you've got a basic research project and you know it's quite low in the TRL, and you're going to apply for a funding call and you can see that it's translational, you need to really read and see where your research could fit in there, because it may be that from the outset you're not going to be considered because it's not translational. So it's the remit of the call, and you know, if they put certain things in there, some of the questions related to technologies that would be applied or could be looking at, you know, for example, certain progress has been made and it's for high income countries, but it's not addressed low to middle income countries. If the remit allows that, then that's fine. But if it doesn't, you know, you have to be careful not to waste your time. When you apply for translational funding, it's very competitive. It's almost always peer reviewed, and that's global peer reviewers. And do ask the question, if you are applying to a broader call and you are having something that's going to apply to low to middle income countries, that the reviewers understand that, because the reviewers will look through it. If they choose only reviewers who do not understand those regions, they will look through it with the lens of someone who's applying it to a high income country, and they may turn around and say it's already been done, where they've missed the point of what you're trying to articulate. So it's important that you do read the criteria, you do ask questions if it's not there. Have a look at FAQ the FAQ sections, as well as what they put clearly, and also have a look at what they'll fund and what they won't. Because the thing is, you could be caught up by something quite small. So it's making sure that you have that. I mentioned proof of concept, and I've mentioned it several times because it's key for translational research. And it doesn't mean, and this answers a question that was earlier, do you have to have done the basic research? If you are working and it's translational research, you don't have to have been the person who personally conducted the basic research, but the basic research or the basic information would have to have been done somewhere and published, because that's what you would use to support your concept. Some of that you may repeat, because as we mentioned before, you're moving on a bit and it has to be right in your hands. But also what you're introducing, if it's sometimes just some other way of doing something, you need to have that comparison. How does yours measure against this? Or what happens in this, you know, scenario? You have to almost put your mind, you know, you have to think strategically. When you're applying for something like this, you have to think strategically, why should your proposal be supported over something else? Also, why should, you know, what questions would they ask? What are the possible pitfalls? Make sure those are addressed. If you haven't done the research to address them, make sure the research that you're proposing will address it, and in a timely fashion, not later on, because they won't fund something that I'll look at that later, because it has to be systematic and incremental. It can't be that you do a massive experiment where it's groundless because some of the key fundamentals have never been. You won't get the support for it. So it's really making sure that everything, the data, everything is robust and it makes sense. You know, it's great having highfalutin concept, but the thing is, like, when it comes to this, people have to be able to understand it and believe in it. We've touched on the key aspects, I think, that will help feed into any translational research proposal that you may have. Key elements of a strong translational research proposal. The next, because I've only got like 2 more slides here. One is this one I'm going to talk through. The other is a wish list, because as I mentioned before, LifeArc funds translational research. So it's really providing insight of what we would look for, some key high level. Obviously, it's very specific depending on what your proposal is. So there'll be some key things related to that. But almost every translational proposal, you'll be looking at some of these things, and it should have these and some. So first is unmet medical need. And this essentially means that if you're saying a population or an indication needs to be addressed because there's no cure for anything, there's no treatment, extend your life by 6 months, and it's quality is low, that's an unmet medical need. If there is platinum level therapy for something that cures it and is available globally and is accessible, then there isn't an unmet need. If, for example, there is a cure, it's available only to some patients, it's not affordable, then there is for a particular setting, but overall there isn't an unmet need. But the thing is, there is in those settings that really would not have the millions to pay for this because it's not available to everyone. So be clear.
On that, Anji, can I just ask, so some of the questions that it came on this training program, especially on the previous session that was about grants and
Yeah.
basic research grant funding.
Yeah.
Some of the guys here, so they are working on things that have been, proven elsewhere, and they are applying the same knowledge on their countries.
Yeah.
Could they use something like what you just said to sell their idea, to say, for example, for cancer, to cure a patient with cancer, you need X, Y, and Z, and it costs whatever.
Yeah.
For us here, it's really hard, for example, to get access to X, Y, and Z meds. So this is our proposal to overcome that. Would that be considered something like that?
Yes, you can, but there is a catch, because the one thing that may stop you is intellectual property.
I don't know. Are you going to touch on that?
I'll touch on it a little bit here, but because it's such a big area, it's almost something that lends itself to a session by itself.
Yeah, perhaps we can leave it to Q&A, but yeah.
Yeah, but that's the one thing that could catch you out, because intellectual property, high level, this is the rights by someone who has discovered something. When they register it and disclose it through a patent or whatever they've used, maybe trademark or whatever. And most of the time, for technologies, it's a patent and design rights for diagnostics. But the thing is, it is where they would basically say who can use their technology. They can say you can't, they have a monopoly for 20 years from the data filing for a patent that someone else cannot use it without getting a license. And that license, they can charge what they see fit. And usually it's to recoup the cost of their research to bring it to that stage. And as I mentioned before, to get, for example, therapeutic, a small molecule to market could be 1.3 billion. So can you imagine the licenses for hit technologies where potentially it could be global? That's the one thing that could catch you out. There are, cases where extreme emergencies where you could have, the government comes in, and for emergencies, they can do something like, for example, COVID, but they can manufacture, but that's only for that particular case. It wouldn't be, those are unprecedented circumstances when they'd be able to do that. So this is the one problem that you may have that may restrict you. And it's checking. What you can do is you can check where the patents are, if there's a patent in that country, which is fine. But it may be that your approach or your solution is different anyway. So then that's not an issue. But it is where the fact that it addresses a population that would not be addressed normally, it is an unmet need, and it is something that you can apply. And there are funders that, not that many, but the thing is there are funders that will support research that does that. Because if you've got where repurposing or something quite simple could treat, you know, an indication that it's not even an issue or no longer is an issue in high income countries, why wouldn't you do that? So key elements of a strong translational research proposal: unmet medical need, which I've mentioned before, the background, and I can't stress this enough, because the thing is, when you read sometimes a proposal, it looks great. You know, when you start reading it, you realize they haven't done their research. They haven't looked. As soon as you start looking around, you realize, wait a minute, they haven't taken into account this technology or this advancement. How comes they're saying that? And it's basically tunnel vision. You can't do that. You have to understand the landscape. So, you know, you hear the term landscape analysis. It's because you're looking at who else is published or who else is technology on the market that covers that indication or similar. And this is where it gets difficult, because the devil's in the detail. Sometimes someone is highlighted where they can control this particular pathway, but it's applicable to other diseases as well. It's just that they haven't, that application has not been developed as far yet. So it's looking and making sure that you're not treading on someone's grounds, because you were either IP or the reviewer is saying, like, this is already being done. How does it compare? You'll be on their tails. That's something you need to consider. And it could be that it's already being done in low and middle income countries. So it's being aware, and that's really literature review, looking around and really making sure that you understand what you are addressing. The team for translational research, I mentioned that it's a collaborative approach. So you have, you know, you can't do it by yourself. You will need to have expertise coming in together. The further it goes along the development pathway, the more expertise you need to have, because the closer you get to the clinic, the more insight you need to have on how applicable this will be for a patient, but also what would it look like really as a therapeutic or a diagnostic, or in the real, what would the product really look like that would cure this disease or manage this disease or whatever it's planning to do. And the only way you can do that is by speaking to the clinicians and have clinical insight. But also sometimes it's where you need to have a specialist that works viruses, because that's what's going to deliver, or, you know, a particular pediatrics, if it's get for young population, or even if it's both, you need to start bringing in immunologists, because it involves a lot of, it could potentially hit on the immune system, or you need to dampen the immune system to treat. And then hopefully you need to bring in that expertise. But also even simple things. For example, you're going to be using particular techniques. You mentioned, you know, next generation sequencing, but you don't have any next generation sequencing expertise there in your team. When you're putting a proposal through and you're saying you're going to use it, they're going to want to know how feasible that is, and also the cost that's incurred. When you're talking about the latest PCR, it's great using, you know, you say you're going to use CRISPR and it's like a buzzword, but in reality, how useful is that going to be for what you need to prove? It's making sure that the resources and the team are there, and where you can, you collaborate, because that saves money, time, but also it saves face when the work that you put in to apply for something that's really not going to be supported. Intellectual property, I mentioned before. Make sure that if it is something new, you think about a strategy, how you're going to protect it. It doesn't matter the territory that you have, there should still be a strategy. Because if you don't, you know, someone else could well before you. And also when you're going to publish, if you need to publish, are there restrictions on that with the funder? Or does the funder want to see certain IP in place? Or translational there will always be that they want to see a strategy and how this is going to be managed, whether it's going to be trade secret or it's really, and have you got freedom to operate? This is the term that's used. And this means that you can do your research up to a certain point, but if you really are going to be getting this to the patient, if there's any sort of proprietary rights or you need to get a license from anyone else, do you know if you could get it? The material that you're using, do you have the appropriate consensus for materials transfer agreements? It's really looking at all these practicalities, like can you do it? And is it feasible? I mentioned competitive landscape before, and this is really like, is there someone else working on the same thing at the same stage or just a little bit ahead of you? It's really trying to do your homework and find out. It's difficult, but it does need to be looked at, because the shock sometimes is when you have something that's really lovely, proposal, project, real need and everything, you submit it and it comes back by multiple reviewers saying like, oh, but this is being done here and it's under this grant or whatever. So it's not only looking at what's published, it's looking at clinical trials, looking at, sometimes people publish like a non-confidential when they get a grant. Have a look there to see, can you see anything that really overlaps with what you're proposing? And how much of a problem is that going to be? But think strategically, why would a funder give you that money if this is happening? But probe further anyway. You know, you can speak to, most funders are quite approachable before you submit. So you can have a conversation, because it's a lot of work to put in a translational funding application for anything. I mentioned before the development pathway. It's key that you understand that and what the steps are and where you sit within them, but also how far you can take it and whether it would be attractive for someone else to take further once you've gone as far as you can. Yeah. So this last slide is basically what I'd be looking for, what LifeArc would be looking for in a translational research proposal. If we're looking just high level, just not very specific and particular indication, we would be looking at, first of all, and sometimes this is even before they've completed anything, because a lot of the time what we do is when you apply for our translational fund, you would send a non-confidential one-page outlining what your proposal sort of area is. And from that, we may have a meeting to discuss really what you're planning to do. And I'm really looking for these, but an indication that these are going to be there, but also that it falls within the funding brief. Because if it doesn't, you can't take it any further. It's not appropriate to take it further and waste your time or mine. So the first thing is making sure that it falls within the brief for that fund. Also, one of the things is that you've got, when you have a meeting with them in the non-confidential, it's fine. It looks as though, because it's very high level, it looks as though it's going to fall within the brief. But the thing is, you're just thinking, is that a real need? What's the size of the population? Unless the brief says it's rare, or, you know, and it should be the classified rare where it's 1 in 200,000, or it's super rare or whatever. Who's gonna need this and who is gonna use it? So it goes back to like that question with regards to technologies that only apply to high-income countries, but they haven't really thought about the low middle income. If the funder really wants to make sure that it's everywhere, they will tell you that it has to, otherwise it doesn't quite fit within the brief. So make sure that you can see it fits within the real world. Visibility is, again, that taps on some of the practicals. Expertise within the team. Say, for example, you're an immunologist, but you're proposing a gene therapy project and no one in your team has that skill. Or you're in an establishment where they don't have, you know, some of the key apparatus and things like that. How are you going to get it? Particularly if, you know, you've got, unless it's specialist equipment, it's not supported. Who's going to fund all these, and how are you going to bring that in to deliver and compete with someone who's been in that field for ages? So making sure the team and resources and everything is feasible within the time frame. Can you really do that? Translational researchers, proposals usually have a Gantt chart showing times, because it's milestoned also a lot of the time. The funding is milestoned, and this is to manage risk, mitigate risk, and also funding, but also it's really to make sure that it's research still, even though it's translational, anything, it can stop, where it's not just you go off and do whatever, and you don't have academic freedom to do what just you please to do. Make sure that you understand the path, because I would be looking to make sure that when there's a question, usually an application has questions, and some of the questions are looking to probe these. So for example, how would you see this get into market, and in what time frame, and who would do it? Have you had any conversations with whoever would take it on the next stage, or would you collaborate with them? So it's understanding the path to the patient. IP, big area, but usually what you would do is, most universities a lot of the time will have either a person or a department where they can bring in expertise to get advice on having an IP strategy. Some point, if it's novel and it fits all the other criteria, you can file a patent, but you don't want to do that too early. But you would have a strategy that when you do have enough data and at the right time, and if all the ducks are in a row, so it looks as though it's going the way it is, that would be the next stage. But also it's a way of ensuring that that technology gets to the market and not someone just really stops you, either freezes it or uses it somewhere else, and then you're blocked out from taking it to the market that you want. And I think this is quite key for low and middle income countries, because it is where IP is the hidden challenge that, everything else could look fine, but you can't get it there because someone has patent rights, but they're not exercising it in that country, but they do have it, and it will stop you doing anything commercial because you'd have to get a license from them. So it's having a strategy and having a way to work around it. Is what you're proposing clear, logical, and practical? And this is just basically, sometimes it could be the way it's articulated in the document. So how it's written. Can I understand your justification, your hypothesis, the data that you have to prove that so far? But if you jump from one place to another, it's not going to sit well. The reviewers will destroy it when it's sent out to them, because they'll just say, like, too many gaps, it doesn't make sense. And they'll critique it, and it will be quite severe, but it's a learning experience to understand that they need to be able to follow it. But also sometimes if it's a stage process, which it can be with translational research, so it will be like first, so expression of interest, then you may have maybe 3 rounds where you have like a smaller application, then they ask for more details. You may not get past that first or second round. And that's a lot of work to get there and a lot of time. With some of the funds that I was working on before with my previous employer, it was 9 months from the beginning right to the end, the funding panel decision. So, but at the end of that, those applications were over 200 pages, because that's the level of detail that they want. And with some technologies and some translational research, it can be like that. The funders, when you go through with the level of detail that they want, it can be something like that. So you can imagine how competitive that is, because translational research is not cheap, it's quite expensive. Last but not least, enough detail. Don't assume that someone is just going to, you glaze over all the background, or you glaze over certain areas and how things are going to be done. It will stand out with fresh eyes, which is when the person who's looking at it, when it's submitted, will see it straight away. Or they'll be thinking, wait a minute, I don't think they're not going to do any homework. I've seen where applications have proposed things that clearly have been published by them years past, but for some reason they think that you're just not going to notice that. It's just how does that hang with what you're proposing? What's the difference? What's the step forward? It is why you have to be, it's competitive. You just get knocked. It's translational research. There's no room for that, because it's so competitive and so difficult to get funding that you have to make sure that all of these things are in place, because you'd be pounding and working non-stop only to receive rejection on top of rejection. Yeah. Okay, I've covered the 4 sections and I'm hoping that they were clear. Any questions?